Patrick Stover, who is here with his wife, Dr. Denise Stover. Both of them have PhDs in biochemistry from Medical College of Virginia, is it? Yes. Patrick went on to get a postdoc in nutritional sciences at the University of California, Berkeley. I've been here 23 years at the university, 18 years as department head, rubbing elbows with administrators throughout the campus. I can't remember a time, maybe Frank, who's been here longer, can remember a time when a member of the upper administration has given a talk on science. But it's fantastically wonderful to be able to introduce Patrick for such an event. He did come to us from Cornell University, and not to belabor the theme, but of all the candidates who came in and interviewed for the Vice Chancellor and Dean position, his talk started with a brief overview of folate metabolism, and then he went on to talk about his administrative experience. So this man knows what science is all about, and he continues to practice it every day. He's many honors, of course, not the least of which is 2016 elected to the National Academy of Sciences. But both before and after he became a member of that organization, he was very involved and has been involved, continues to be involved in various policy committees at the federal level for dietary recommendations and so forth. And I was at a all-day retreat yesterday. Those of you who know me, you probably don't even have to know me that well to know maybe my attitude going into that retreat. But it was quite refreshing to have a day-long discussion of potential policy and strategic visioning and so forth, where it was clear, without even having to make the point, that science was driving all of the discussions, and he wanted science to drive what the college and AgriLife does for the next several years. So, without further ado, Patrick, the floor is yours. Welcome.
Thank you very much. Thank you for the opportunity to speak here. So I have not changed from my Cornell slides. All of this research was done at Cornell University. I have to move my program here. I can't even think about that to the summer, and my grants. But this is my group as it stands right now. I have funding for both R01 from NICHD, which really relates to some of our mouse models of birth defects, and this is a R37 American award from NIDDK that still has 6 years left on that. So I'll be moving these grants. I have money. I have no time. I have to figure that out. But these are the students I have now. This is a visitor here from Germany who's been doing some of the metabolomics with us. These students come from both either the nutrition doctoral program or the biochemistry molecular cell biology PhD program, and there is from the veterinary college. But the work I'm going to be talking about today is mostly Elena's and Martha's. So first, I just want to introduce you to neural tube defects. You may not think about this every day, but this is one of the more common rare birth defects. And what happens in humans between day 23 and 25, you have rapid proliferation, survival, differentiation, migration of the neural epithelium, where humans go from becoming essentially a two-dimensional organism to a three-dimensional organism as the neural tube folds and you get zipped up the back. This is the head region, this is the cranial region, the loose will come here and here. But during this time between day 23 and 28 is the time of the most rapid cell growth there is. And if there is insufficient rates of these processes as the neural tube closes, within that critical window, it will remain open. And when it remains open in the head, you have something known as anencephaly, which is always lethal because the head is open and the brain doesn't develop. If it happens lower in the spine, you'll have what's called spina bifida, where you have a pore, and then when cells later come down and migrate to innervate the limbs, they migrate out of the pore and you get that mass. This is one of the more common birth defects of the rare birth defects, and extraordinarily expensive. So again, it's one of the more common, rare, congenital birth defects. The rate is there's about 2,500 birth defects each year. This is the rate in developing countries, telling us there's probably some environmental component to it. But the recurrence rate is 1 in 5. So we know there's also a genetic component to this. And we know that this birth defects runs in families. And so the problem is we don't know what the gene variants are, and there haven't been enough births to do a really good comprehensive GWAS to find out what the risk alleles are. So we can't identify these people who are at risk. And the critical period for this happens with a month of fertilization before a woman even knows she's pregnant. So it really closes that window for possible prevention. But what we do know is that clinical trials going back into the 1990s show definitively that 70% of these birth defects could be rescued with maternal folic acid supplementation. So it's a huge public health success story. The problem is we don't know how it works or why it works. I have the wrong version of it. I'm sorry, I just want to quickly load another version.
Are you guys need to do something?
Yes, sir. We were told that the live stream didn't work, so we're here to try to fix that.
And what do you need to do? Now's a good time, because he's cheating.
There we go. Is that the problem?
We did do either, but it was, we probably easier to run it on this. Yes, it's true. You go to WebEx. Website name and setup.
Clinical trials have shown that we can prevent this birth defect. A lot of countries still will not fortify the food supply with folic acid like the U.S. has. And there's several reasons for this. Remember, a clinical trial is where you give an exposure and you look for a clinical outcome. And you don't necessarily have to understand the mechanism. And that creates uncertainty. Other issues with why countries don't want to fortify is because this was really the first fortification initiative. Putting a vitamin in food that didn't seek to remedy a nutritional deficiency. There's no biochemical deficiency really in these individuals, rather than you're seeking a medical purpose, using food as medicine. Very controversial at the time. It exposes everyone, but targets just a small population group. And so there's some people who have different biochemistry. They need more of this nutrient. But the question is, if it's only 2,500 births, you get a small group who's benefiting, or others, is there another small group who might be accruing risk with that? And some of that stems because of the folate-cancer relationship. We know that there are many anti-cancer agents, methotrexate, uracil, permatrexate, that kill cancer cells. Well, the premise is, well, if you give people more folate, is this going to somehow increase cancer risk or make tumor cells grow faster and escape that window of treatment, etc.? So you'll see in the literature a lot of uncertainty, not a lot of evidence, but certainly uncertainty about this. I don't want to get into that. There's other concerns that when you take this level of folic acid, you get unmetabolized folic acid in the blood. And the important point here is folic acid is not a natural vitamin. It is a provitamin. It was developed by Lederle back in the 40s, and the reason they developed it is because it's stable. Folate in food is highly, highly unstable. It oxidizes extraordinarily rapidly. You cook your food, it's gone. This is stable, so you can put it in food, and it will be stable, and you can raise people's folate levels, as we'll see. But all of these factors really exacerbated common concerns of unintended consequences. So you have countries in Europe don't fortify, the U.S. does. But again, one of the big barriers here is understanding how it works, so we can tell whether or not there are any unintended consequences associated with the fortification. So what do we know about folate-related pathologies? What we know for all of them is they are complex traits. That is, there are polygenic diet interactions that give you risk for all of these outcomes that are related to folate nutritional status, how much you consume, how much is in your blood. But we know that folate deficiency isn't enough have risk for these outcomes, you have to have impaired folate status, a nutritional deficiency, or genetic variation that gives you a deficiency. You don't absorb, you don't transport very well, etc. But that's not enough. You also have to have impaired folate utilization. That is, you have to have either some sort of a genetic predisposition or an environmental or other insult that decreases your ability to metabolize folic acid. You need both. The impairment that is usually driven by genetics and the exposure that's usually driven by diet. And when you have these two together, then you have risk. Then when we talk about how do we get between folate deficiency and these disorders, there are two major pathways, as we'll see, that connect folate deficiency to these disorders. One is you need folate to remethylate homocysteine to methionine. Methionine becomes adenylylated to form another cofactor, a methylation cofactor, that is involved in about 150 methylation reactions. Methylation of DNA, RNA, proteins, small neurotransmitters, phospholipids, etc. But you also need folate to make nucleotides, and when you're folate deficient, you have impaired DNA synthesis, lower rates of cell division, elevated uracil in DNA. We'll talk about DNA instability. And both of these happen simultaneously. So when you look at genome type readouts in folate deficiency, everything is moving. Genome stability is moving, methylation is moving, expression profiles are moving. So it's very difficult to determine what the actual pathway is. But one of the fundamental questions in the field has been, is it all about you or me? That is it about uracil and DNA, or is it about methylation? And my wife here, who works in my lab, always tells me, I think everything's about you. Or she says, you think everything's about you. And there may be some truth to that. But a little bit about uracil and DNA, again, as a means of introduction. The T base in DNA is the only base that is non-essential. That is, if you don't have sufficient pools of DTTP, DNA polymerases can't discriminate between T and U, and they will then plug in DUTP. So there's a literature out there, whether it's correct or not, I would argue that the ratio of T to U is what determines the amount of U that is misincorporated into DNA. And what regulates the amount of U in DNA is both the supply of DUTP, rates of excision-based repair that remove uracil from DNA, giving you an A-basic site that then can be repaired with DNA synthesis, or result in more uracil misincorporation. When this ratio is out of balance, you can get successive rounds of incorporation and base excision that lead to strand breaks, excessive repair, and apoptosis. There are enzymes that help prevent this from happening. One is thymidylate synthase, the enzyme that converts uracil to thymidylate, we'll see, and DUTPase that tries to, or does, convert DUTP to DUDP so that it can't be misincorporated into DNA. And so we're very interested in, as you'll see, how these all interact to regulate uracil levels in DNA. So now I just want to get into the fundamental metabolism. So this is a little bit of a scaled down version of folate-mediated low-carbon metabolism and that metabolic pathway. And you see here there were three primary products of the pathway: purines, thymidylate, and these methylation reactions coming from this adenosylmethionine. This THF is tetrahydrofolate. That is the vitamin cofactor. And folate serves to chemically activate and carry one carbon units in the form of formate for one carbon transfer reactions. So the same way that ATP carries activated phosphate, folate carries chemically activated one carbon units for one carbon transfer reaction. So formate in an ATP-dependent reaction condenses onto tetrahydrofolate to give you 10-formyl tetrahydrofolate that becomes the number 2 and 8 carbon in the purine ring. You can reduce that formate group to formaldehyde, giving you methylene tetrahydrofolate that can serve one of two fates. You can reduce that one carbon again with NADPH to methanol to give you 5-methyl tetrahydrofolate, 3-methylate homocysteine and methionine. Methionine can go into protein or become adenylylated, give you adenosylmethionine that can methylate again up to 150 different molecules, giving you spent homocysteine that can be remethylated. Or alternatively, methylene tetrahydrofolate can be that formaldehyde can be incorporated into DUMP to make thiamidolate eventually. So this is pretty simple. You have a metabolic network, create products. What we are interested in our lab is how decisions are made at various branch points within this network. And one of the most critical branch points is this one. And what's critical to consider in this pathway is that in any tissue or cell, if you calculate the total number of folate binding sites, enzymes that bind folate, and compare that to the amount of cofactors, there's somewhere between 5 and 10 times more enzyme than there is folate. Cells never saturate with this cofactor. So therefore, all of the pathways in this interconnected network are competing for a limited resource, the amount of cofactor. So these decisions become critical. One of the questions that was really driving what we wanted to know is in folate-related disease, how is this decision managed? When you have a limited resource, how do you decide whether methylene tetrahydrofolate goes to methylation or goes to make thiamidolate? How is that decision made? Simple kinetic modeling that was done decades ago said, well, if you just build a simple kinetic model, what you will see is that this pathway is preferred, especially in folate deficiency. But that's not always what the actual experimental evidence shows when you test these things. So we were interested in really instill how that decision is made. So one of the ways we begin to approach that is to look at sources of one carbons. So there's two ways you can get to this molecule. One is to take formate, condense it, tetrahydrofolate through the enzyme MTHFD1 we'll talk about, and then reduce that one carbon to formaldehyde. Alternatively, you can take tetrahydrofolate and serine to glycine and make methylene tetrahydrofolate directly. So there's two sources, serine and formate, to make methylene tetrahydrofolate. So one of the questions we wanted to ask, well, does it matter what this one source is? Does that affect how that cofactor there is partitioned between the two pathways? And the way we did that as experiment is we put in serine that was labeled with 2 deuteriums in the hydroxymethyl group. Serine then goes into the mitochondria through mitochondrial one-carbon metabolism through the enzyme serine hydroxymethyltransferase 2, an isozyme of 1 encoded on a different gene, eventually forms formate, and then it can go down to methylene tetrahydrofolate. So D2 serine becomes D1 formate, but of course the D2 serine, if it goes in this direction, is going to give you D2, 1 carbon with 2 deuteriums, that'll give you a 1 carbon with 1 deuterium. Then we can look at DNA and we can look at protein and look at those 1 carbons and ask the question, what is the relative ratio of D2 to D1? That is there any difference in the way those 2 carbons derived from different sources partition into those 2 sinks, if you will, and the fact is they partition differently. What we see is thiamidolate prefers to get its 1 carbon D2, prefers to get it from serine hydroxymethyltransferase, whereas methionine tends to prefer to have it from formate. And we see this in every single cell that we look at. The numbers are different, but it's generally about a 2-to-1 ratio. So this tells us that this isn't right, that there must be some sort of compartmentation or something that allows these 1 carbons to preferentially go this way and these 1 carbons to preferentially go that way. So we wanted to begin to understand this, and we wanted to understand this partitioning in the context of folate-related pathologies, especially neural tube defects. So this is just a different version of this, just the mitochondria are built out here. So mitochondria with these folate enzymes take serine or glycine to catalyze it to formate, and those are the 3 reactions there. What we discovered is trying to understand this partitioning is we realized, well, there must be 2 pools of methylene tetrahydrofolate. One that comes from formate, one that comes from SHMT2, but they must exchange to some degree, right? Where the SHMT2 derived 1 carbons are preferentially going to thiamidolate, these are preferentially going this way, but they can exchange. So we began to look at the characteristics of this pathway and trying to see whether or not there was some sort of a multi-enzyme complex or something that would allow that preferential distribution of the 1 carbon. And through those studies, I'm sorry, I'm going to go into one more point here. Through studies where we manipulated SHMT1 in both cells and animals, we knew that SHMT preferentially supports this pathway, but it supports both. SHMT expression determines thymidylate synthesis capacity. That is, if you overexpress or knock down SHMT1 in any cell line, you will get proportional changes in rates of thymidylate biosynthesis in cells. It's almost 1-to-1, which we had to explain. We went on to express a mutated form of SHMT1 where we mutated the active site so it had absolutely no activity, and it had exactly the same effect. That is, rates of thymidylate biosynthesis increased even if you expressed a mutant form with no activity. But of course, formate is still the major source for both pathways. So we had to explain all of this in whatever sort of mechanism we derived. Well, Colin Waller, who was a BMCB student, biochemistry student, who's now faculty at University of Rochester, was investigating whether or not there was some sort of multi-enzyme complex, and what he discovered that all three of these enzymes were modified by SUMO. SUMO is a polypeptide, it is a cousin of ubiquitin, and when proteins are sumoated, they are then targeted for nuclear import. So in fact, there was evidence that this pathway here actually translocates into the nucleus. This would explain why we got preferential enrichment of the SHMT-derived 1-carbons in the thymidylate, because this pathway removed itself from the network and went into a different compartment. Still didn't explain how formate got into this pathway, but we were beginning now to understand this competition was actually not a kinetic competition, but had to do with changes in cellular compartmentation. At the same time, we were doing mouse studies, and the whole field was doing mouse studies, asking the question, within this network that is tightly connected, you take any perturbation anywhere in this network, you'll affect all of these readouts. Which one of these are responsible for neural tube defects? And we have a lot of data on this now. Both of these genes have been knocked out. When you knock out these genes in mice, you get neural tube defects. That's really, really satisfying, because it tells you if you're folate deficient, you can get a neural tube defect, or 1-carbon deficient, if you will, you get a neural tube defect. The problem is they're not folic acid responsive. And 70 to 80% of human NTDs respond to dietary folate. These don't. So it's interesting in terms of mechanism, but it doesn't relate to the human condition, where most of them are folate responsive. Purines have really been understudied. This pathway here has been knocked out, overexpressed, all of these enzymes manipulated. No one has had a neural tube defect that had occurred in isolation of just catastrophic developmental defects. And so therefore, this was the bias in the field that neural tube defects were probably a methylation defect. There's no evidence for that. We created a mouse where we knocked out SHMT1, and in fact, we got folic acid responsive neural tube defects. As you can see, we could get it with just deleting 1 copy of SHMT. And there's a long clinical literature that antifolates like methotrexate or 5-fluorouracil cause neural tube defects. So now we really wanted to understand nuclear 1-carbon metabolism. Why does this pathway go into the nucleus, and how does it affect risk for birth defects? So we did a number of studies to try to identify where these enzymes were in the nucleus. I'm just showing you one here where we took the de novo thymidylate synthesis pathway here. Here's thymidylate synthase, here's SHMT, dihydrofolate reductase. They take tetrahydrofolate to an activated 1-carbon form. Thymidylate synthase takes D1P to thymidylate, giving you a spent cofactors dihydrofolate, which has to be re-reduced to tetrahydrofolate. That's the pathway. We had evidence that was in the nucleus. It was stimulated. Here you see if you express thymidylate synthase as a GFP fusion, you see it all over the cell. It's overexpressed. But in the nucleus, you see it there as filaments. If you do immunolocalization with S-phase flock cells, you'll just see it in the nucleus in these filaments. There's SHMT1 as a yellow fluorescent protein. Again, as a filament in the nucleus, lamin 1, the nuclear filament. Overlay those, and you can see this pathway exists as a structured complex in the nucleus, tightly bound to the nuclear lamina. We verified this by immunoprecipitation reactions and also showed that sumolation was essential to keep this complex together, the red dot being the sumo residues. So sumo is the Velcro that keeps this complex together. We also showed that SHMT binds very, very tightly to the nuclear lamina. It is the scaffold that keeps this complex together. When that enzyme is knocked down or deleted, these enzymes, as you'll see, become diffuse. And that's shown here, where you have siRNA against SHMT1 and 2. You can see we were able to knock those down effectively. You can see in a MOX transfected cell, thymidylate synthase, the green protein, is there and it's structured. When you knock out SHMT1 and or SHMT2, it becomes completely diffuse, and you no longer have activity in the nucleus. So this pathway exists in the nucleus, and it exists there as a metabolic complex, and that complex is required for activity. And what we know with the mouse model, again, is that if we disrupt SHMT, and in the mouse, we only have to reduce activity 30 to 50%. The only biomarker we see associated with that is elevated uracil DNA. There is no change in serum biomarkers related to 1-carbon metabolism or any other biomarker for that regard. We see this elevation in uracil and DNA. We see more gamma H2AX, evidence of DNA repair and strain breaks going on, but it gives us exencephaly. And the penetrance is about 10 to 25% depending on the inbred mouse strain. And they recapitulate the human condition. These neural tube defects only happen when you restrict folic acid from the diet. They have to be folate deficient. You have to have an impairment in thymidylate biosynthesis by this level. And again, the risk is associated with a really subtle alteration in metabolism, the readout here, which is elevated uracil and DNA. Just an example, in these mice, the uracil levels in DNA between a wild-type mouse and a hectare in liver is about 3 to 4-fold. This is how much it's elevated in an APC-min mouse in the colon. And you can see here we can do it under folate sufficient or deficient conditions. And we have plenty of folate in the diet here. But you knock out one copy of the gene, about a 3-fold increase. That's exacerbated with folate deficiency. So again, uracil and DNA is the only phenotype that we see in terms of the molecular phenotype in neural tube defects is the pathological outcome. So one of the questions we wanted to ask them, well, what else is in this complex? Is it just this pathway, and is it in any way associated with the DNA replication machinery? So we did tandem affinity purification using SHMT1 as our anchor there, and in fact, we pulled down the entire DNA replication machinery. We also pulled down cell cycle proteins, we pulled down lamin binding proteins. Of course, you would expect this. SHMT1 binds to lamin, so you're going to get all the lamin proteins when you do that. We also found the de novo thymidylate biosynthesis pathway, including, importantly, MTHFD1. That was not an enzyme we initially thought was in there, but this is the enzyme that brings formate into the 1-carbon pool to give you that D1 thymidylate. We also pulled down KDM1, which is a histone demethylase that has been shown to be folate dependent. So we wanted to ask the question, does this pathway actually ride with the DNA replication machinery during DNA replication? So we transfected cells, or actually stable cell lines, expressing an artificial chromosome here that had a defined origin of replication. This is a well-studied system, and we know that during DNA replication, about half of the replication machinery is sort of stalled and waiting at the origin of replication. The rest is scattered throughout the plasmid. And in fact, when we did CHIP studies, we found that half of the SHMT was at the origin of replication, and the rest of it then, at about a level of about 20%, was scattered around the rest of the chromosome. So this again for us was good evidence that in fact this pathway, this biosynthetic pathway that makes thymidylate rides with the replication fork. And that T is the only base that's made sort of on-site, on demand. All the other nucleotides are made in the cytoplasm and transported in. This one is made on-site and on demand. What's interesting to us too is yeast don't do this. Yeast don't simulate the pathway. Yeast don't import the pathway into the nucleus. This is unique to mammals. And so one of the questions we were asking is, well, why? Why do you need to make thymidylate on-site, on demand during DNA replication? I want to focus on MTHFD1. So this is the enzyme that came out of this mass spec purification that we did of the complex. And we first wanted to see whether MTHFD1 did localize with SHMT, and we did this through immunoprecipitation, but these pictures are prettier. Here you see S-phase block cells, and what you see here is a green fluorescent protein of MTHFD1, and you see here it's in these beautiful filaments here in the nucleus. If you knock down SHMT1, it becomes completely diffuse again. So this enzyme is anchored to SHMT as well. This again explains the importance of SHMT1 in terms of neural tube defects, and why as you overexpress it or underexpress it, you get similar increases in rates of de novo thymidylate biosynthesis, because it's the anchor for the scaffold. It is the scaffold that determines how many units of that metabolic complex that you have at your disposal. So this addressed another interesting question for us. Because initially, remember, we were asking the question, how do you decide if this one carbon goes this way or this way, right? But then we realized, well, this is compartmentalized, so it's really a faux choice. What the real decision is does this enzyme, what determines whether this enzyme goes this way into the nucleus or stays here? Because if this enzyme partitions mostly into the cytoplasm, you're going to support methylation reactions. If this enzyme, again, partitions into the nucleus, you're going to support thymidylate biosynthesis. So how is that decision made as to how this enzyme partitions between the cytoplasm and the nucleus? And what happens in folate deficiency when we believe that this pathway is prioritized over this way? And so we looked at some mice we had, and these are gene trap mice where we knocked out one allele. It turns out it doesn't even matter. This could be any mouse. But if we look at a liver, and we look at whole cell extract, and we looked at this is how much MTHFD1 there is when the mice are fed this diet that has plenty of folate. There's that much enzyme in the liver. The gene trap, there's less. If we remove folate from the diet, we make them folate deficient. Not very much change in terms of protein levels. But look at nuclear localization. If we look at how much MTHFD is in the nucleus, we see now that in folate deficiency, this enzyme just packs into the nucleus. So this is how the cell prioritizes when folate becomes limiting, whether it wants to support thymidylate biosynthesis or homocysteine remethylation and methylation reactions. It clearly votes, we're going to put this in the nucleus, and we're going to ensure rates of de novo thymidylate biosynthesis are sustained. At the same time, we received fibroblasts from a patient at McGill. So this was a patient who was presented with failure to thrive, as well as elevated homocysteine, megaloblastic anemia, and SCID. This was a newborn. They did exon capture sequencing and found that this unfortunate individual acquired 2 bad copies of this gene. The copy from mom had a deletion here in the active site for the dehydrogenase cyclohydrolase domain of the protein. Dad had a mutation in the NADPH binding site that raised the KM a couple orders of magnitude. And so this is 2 sample fibroblast cell lines. This is the affected child, and you can see it had half the amount of protein and only had 1 complete allele, and that allele was mutated. When we studied fibroblasts from these patients, we had 2 control fibroblasts, and then the the proband, the affected child, you can see they are not very good at making de novo thymidylate biosynthesis. This was our isotope tracer study that measures rates of de novo synthesis of thymidylate as a function of the salvage pathway capacity. They don't make it very well. They're fine at making purines. So de novo thymidylate biosynthesis capacity in this child was reduced by about 90%. That explains the skid. De novo purine biosynthesis was unaffected. But when we went back and did our isotope tracer studies with this, that explained the D1 and D2. Remember, D1 means the one carbon came from formate, D2 means it came from serine. What you see here is these are the two controls. That's the D1 over D1 plus D2 ratio. You look at the affected patient, 1/10 the amount of D1. They can't incorporate formate into the one carbon pool, right? They're not very good at that for methionine. But look at thymidylate. It's only decreased about twofold. Why is that? Well, again, if you look at localization here, and this is immunolocalization here, if you have an S-phase block control cell, you can see MTHFD1 in both compartments. Look at the affected sibling. It knows it can't make thymidylate, and it is packing all the mutated protein it can into the nucleus to try to sustain this. And so clearly cells make a choice here, which way they want to send that one carbon, that limiting resource, and under conditions of both folate deficiency or even mutant protein, they try to rescue de novo thymidylate biosynthesis. We're trying to understand the mechanism for this, but we're very interested in the possibility that this protein is actually formulated and have some evidence for that. And that's what helps direct where it goes by competing with sumylation. So that story continues, but what we know is that the partitioning of this enzyme between these two compartments is regulated, and it is critical in making that decision of how you want to spend your scarce resources, to make DNA or to have sufficient methylation capacity. We continue to try to understand uracil in DNA. Again, de novo thymidylate biosynthesis is only localized to the nucleus in higher eukaryotes. Yeast don't do it. We want to know what that is. Now, DNA uracil levels vary among tissues. We want to know why that is. Some cells don't accumulate very much. Some cells accumulate a lot. There's also a deep clinical literature that shows that there are fragile sites within chromosomes that break in folate deficiency. These are known, we know where these breaks are. They're used oftentimes to diagnose specific rare inborn errors in metabolism. This suggests to us, well, maybe uracil incorporation isn't a random event. Maybe the reason that you make thymidylate right at sites of DNA replication is because it gives you a choice. I can put a T or I can put in a U, and that has some sort of a regulatory effect. So we're trying to understand whether in eukaryotes, mammals specifically, whether uracil misincorporation in the DNA is random or not random. That is, there's a choice, and certain sites of the genome are enriched in uracil, and that serves some sort of information or signaling role. And so we're trying to understand that, but we think that this is a good premise to begin to investigate that. We're not very good at this because we try to do uracil sequencing and all kinds of stuff, and we still don't have the technology right to be able to ask that question. So where we are now in our understanding is this is DNA, this is the de novo thymidylate synthesis pathway, we know it's here. This pathway, SHMT, is the anchor and the most important enzyme to give you the capacity to convert DUMP to thymidylate. MTHFD1 is supplying the one carbon in the form of formate for that one carbon. If this pathway here does not function at a sufficient rate, DUMP will be converted to DUTP and uracil will be misincorporated. So again, we're trying to understand this, how it's regulated, what it's doing. But at the same time, we also are doing mouse work. And we want to ask the question, there are many different origins of neural tube defects. Folate deficiency in genetically sensitized individuals is one of the sources of neural tube defects. But vitamin B12 deficiency is associated with neural tube defects. arsenic exposure is, phenomicin is, type 2 diabetes. We're asking the question now, do all of these sort of conditions feed into this common pathway that gives you a neural tube defect? And I want to talk a little bit about arsenic. We were trying to purify a sumolated form of MTHFD1. So we expressed a histide version of this in mammalian cells, and we were going to purify this on nickel beads. Well, the problem was we couldn't elute it from the nickel beads, no matter what we tried. Imidazole didn't work, nothing worked. So I said, well, throw arsenic at it. Maybe it just really likes the metal. And in fact, we got off not only the histide protein, but the non-histide endogenous protein. This protein binds arsenic or metals like a rock. And so you can't use histides with this protein, but it thought, well, maybe the arsenic-induced neural tube defects target this protein. I don't need to tell you, arsenic's a big problem. It's in water, it's in wine, it's in rice, and people care about it. And there's a famous play, Arsenic and Old Lace. And of course, in nutrition, you always have the problem that it used to be offered as a supplement to have very nice complexion here as well, so they used to feed it to people. But we began to look at the interaction of arsenic with MTHFD1. First, we looked at an in vitro simulation kit where we just took MTHFD1 protein, added the E1 ligase and conjugase that modifies the protein with sumo, put in a sumo mutant as a control, and plus and minus arsenic. And what you see here is the presence of arsenic at relatively low dose, which you find in contaminated water, hyper-sumolated MTHFD1 in vitro. And if you looked in cells, these were MCF7 cells, I believe, that were treated with 5 micromolar arsenic, and you look at MTHFD1, you see this smears the sumolated protein here on the western blot. You add arsenic, you get an increase in 1 hour. After 4 hours, it begins to disappear because proteins that are hyposumolated are consumed by the proteasome, and I'll show you that in a moment. But what we found, in fact, is that when we treated cells with arsenic, and we measured uracil and DNA, arsenic induces uracil levels in DNA. And this is probably the mechanism whereby arsenic is known to cause genome instability. There's really no good evidence for that. If you look here at rates of de novo thymidylate biosynthesis using the isotope tracer studies we do, again, this is in cultured cells, 0.5 micromolar, 1 micromolar, 2 micromolar, well within the exposure range, you see dose response. Note this does not saturate. We know now that this is because we're inhibiting the nuclear import, and this is the residual that's going on in the cytoplasm. At least that's the theory. So we know that arsenic, uracil is elevated, de novo thymidylate biosynthesis is depressed. It depletes levels of both MTHFD1 and SHMT1. These are Western blots. This is time after exposure to arsenic. You see the protein levels go down over 24 hours. If you add an inhibitor of the proteasome here, MG132, you don't lose the protein. So in fact, arsenic is encouraging the degradation of this protein. If you look at here, de novo purine biosynthesis, it's unaffected. It's not happening in the nucleus. It doesn't require the simulation. And so one of the proxies that we're beginning to use for uracil and DNA, because the measurement for this is not easy. You have to take recombinant UNG, you have to purify DNA, you incubate it, you cut it out, you measure it and quantify it by mass spec. But we know that when there's double strand breaks related to uracil, you have gamma H2AX, that histone that then associates with those strand breaks. And so we've been looking at strain breaks as a function of arsenic. These are just MCF7s, or these are MEFs here, lacking SHMT in this case. But here's cells grown with folate, no strain breaks, add arsenic strain breaks, make them folate deficient strain breaks, add them together, and you get this huge synergistic effect. So in fact, folate and arsenic interact to affect genome stability. And we can also do this by flow cytometry. We can look here at gamma H2AX intensity as a function of arsenic. You have folate in the media. The dose response, you really don't see it about 4 micromolar. No folate, you see it much earlier. Okay, so that led to the question then, can you rescue arsenic-induced NTDs with folate? right? Maybe then we could overcome this block. And so we did this complicated study. We took wild-type mice, and we took our deficient mice that have, of SHMT1, that have neural tube defects, plus and minus folate, looked at the phenotype exencephaly, and the bottom line here is we see nice dose response between rates of neural tube defects as a function of arsenic, looking at the outcomes, either total NTDs or NTDs per liter, but they were not folic acid responsive. So even though arsenic is targeting de novo chromatolate biosynthesis, it's destroying the enzyme, so adding more folate doesn't help in that point, the input. You can't rescue. And this, we believe, we're publishing this. Now, it's going to be important because the World Health Organization is considering adding folate to the food supply in places where there are high arsenic to decrease rates of neural tube defects. It's not probably going to work. We also did metabolomics. I'm going to skip over that. So, in summary here, we know that arsenic impairs de novo chromatolate synthesis, elevates uracil DNA by affecting simulation. The arsenic-induced DNA damages folic acid responsive in culture cells, but the neural tube defects are not. So this really was a question to us because you may have gotten the sense, our underlying hypothesis is that if you have excessive genome damage, strand breaks, that's what's causing the neural tube defects. Well, here, we can rescue the genome damage, but we can't rescue the neural tube defects. And so that made us a little worried about our hypothesis, but then we began to think, well, what about dihydrofolate reductase? This is the third enzyme in the pathway to make de novo chromatolate. Was it sensitive to arsenic in terms of its levels? And absolutely it was. So these are just maps grown in with different types of folate. These are the protein levels. When you add arsenic here, plus arsenic minus arsenic, the protein disappears. So they may not even be able to use the folic acid, right? Because the enzyme that converts folic acid to tetrahydrofolate is missing. So this is the way the folic acid gets into the cofactor pool. The hydrofolate reductase reduces it twice to give you tetrahydrofolate. So again, we use folic acid because it's stable. The other forms I showed you are not stable. They're very labile. You put them in a test tube, they're there for about 2 minutes and they're gone. So we knew that the redox sensitive site here is this bridgehead carbon here and this bond right here. And so what we wanted to do is we thought, well, can we block that? Can we put a chemical modification here to make a reduced form of folate that was actually stable? So what we took was 5-methyl tetrahydrofolate that is normally unstable, but we put a formate group there. We took 5-formyl tetrahydrofolate and put a formate on N10 to make 5-formyl 10-formyl and 5-methyl 10-formyl. So these are novel folates. We've patented these. And they are perfectly stable, and they use a completely different enzyme to form 5-methyl tetrahydrofolate and 5-formyl tetrahydrofolate, which are natural folates. And we tested their stability, my wife did, in chow in mouse pellets, and in fact they are perfectly stable out to 10 weeks, comparing folic acid to one of our synthetic folates. And they behave like natural folates in cell growth studies. So this is doubling time of cells, this is folate concentration. Folic acid here is a terrible folate. Receptors don't bind it very tightly, it's slowly incorporated, and so you have to get high levels of this compound into culture media or into serum to get it to be used. Whereas natural folates, like leucovorin, are effective at very low concentrations and lowering the doubling time. Well, so are our synthetic folates. So we are now repeating the neural tube defect studies with arsenic, seeing whether or not these novel stable folates can rescue the arsenic-induced neural tube defect by getting around that dihydrofolate reductase block. So I want to close with some studies that we're doing with Microsoft, and just put on my nutrition hat for a minute. And I was part of this National Academy Committee last year. This report came out a few months ago, which was sort of groundbreaking. Nutrient levels, BRDAs, how much of every nutrient you're supposed to consume every day, are derived by clinical studies where you're dosed with a nutrient. They usually look at some functional biomarker in metabolism, look at the dose response, see where it saturates, and that's how much you should eat. Well, what the federal government said was, well, if diet is causing all of this disease and the $1 trillion a year in healthcare costs, stop setting nutrient recommendations and even food-based recommendations based on biochemistry. Set them on a clinical endpoint. Like was done with folate and neural tube defects. That was the example that was used. Well, of course, this is crazy, because no one nutrient relates to a chronic disease. The folate in birth defects is rare. These are all complex interactions of multiple nutrients, multiple pathways that lead to disease. Nonetheless, this was the mandate. And so what this means is now when we consider how much of an individual given nutrient we need, we have to consider systems over pathways, because it's systems that fail in chronic disease, not individual biochemical pathways. We need integrated biomarkers that report on an entire system output. We need to consider reference intakes, recommendations as ranges instead of point estimates. Because of this, we're looking at network function and network plasticity. And we need to type agent. I don't want to get into that. But to address some of this, we started collaborating with COSBI, which is a Microsoft Institute in Italy, in Trento, in the mountains, in the Alps. You have collaborators, you should have them in nice places. But what we began to do was to recreate an in silico version of this network. And the way we did this is we provided all of the kinetic constants for every enzyme in the pathway. We went back and validated all of them, looked in all of the databases to look at what are the expression ranges at each one of these nodes, and put this in their proprietary, what now is a proprietary software that they've developed that does simulations. So what you have is a differential equation base that then has a simulation overlay, and you can allow this to come to equilibrium. We have now put this together. We begin to publish on some of this, and we have a lot more coming. But this is the way that we can begin to comprehensively do dose-response studies in silico, change multiple parameters, and begin to looking what ranges of requirements are as you insert different genetic variants into this or different disease states into the model. So I'll just show you one example of what we did. One of the common polymorphisms you find in folate metabolism is in MTHFR, methylene tetrahydrofolate reductase. This is the enzyme right at that branch point that sends the methylene tetrahydrofolate into the methylation cycle. In this room, 80% of the alleles have a C here, coding for an alanine. Another 20% have a T there with a valine. This protein is thermally labile. And so people here have about, if you're homozygous for this, you have about 40% of the activity that people who are homozygous for this allele. That's a really penetrant polymorphism. If you have this polymorphism, you can't accumulate very much folate, because your folate pools the unstable forms and it turns over, you have a higher folate requirement, and you are at risk for birth defects because of your low folate status. But if you survive here, and there have been multiple epidemiology studies that have done this, if you look at your colon cancer risk as a function of genotype, if you're TT, your colon cancer risk is reduced about 80%. So the same change in biochemistry and affecting that partitioning between DNA synthesis and methylation gives you risk for the birth defect, but potential benefit against colon cancer. So we wanted to understand this more at a biochemical level and begin to model this. Here's MTHFR again, and it's going to be, it's affecting the partitioning here, how many 1 carbon units go here versus here. So the first thing we did in this model is we looked at propensity score as a function of both folate, low folate, high folate, polymorphism, CC or TT. And we looked at propensity scores. And this propensity score just says there was a reaction occurred. There was a firing of an enzyme that happened somewhere within the network. And what you can see here is that as you go from folate replete to low folate, you get more firing, more randomness really is the way that's interpreted in the system. So you get about a 30% difference. If you look at the polymorphism, you still see a folate effect. But if you look at the effect of the polymorphism, you see a 435% difference related to that polymorphism. That is, this network is firing much more when you have that polymorphism. It's more active, You could interpret that as more random, or there's one of the pathways is working at much higher capacity. So through that network, we can begin to break this down, and what you see here is MTHFD1 as a, this is activity, essentially how many firings happen as a function of the MTHFR polymorphism, where we just scale the activity from 2-fold to 0.3, you see its activity goes up. All of the activities associated with that enzyme are going up. MTHFR, well, this is what we're scaling to, so it's going down as predicted. The thymine synthase, the enzyme that remethates homocysteine, follows in parallel. So as MTHFR activity increases, MTHFD1 flux increases. We can look at de novo thymidylate biosynthesis and purine biosynthesis. This is purine biosynthesis unchanged. Thymidylate biosynthesis goes up, as predicted a little bit, but look what happens to SHMT. As you scale for MTHFR, it absolutely takes off in terms of its activity, both the forward and the reverse reaction. And what I didn't tell you is that while these are the two natural substrates, tetrahydrofolate and methylene tetrahydrofolate for SHMT, it binds bimethyl tetrahydrofolate as an inhibitor. And as you can see, as you scale down MTHFR, you make less of this, you have less of the inhibitor bound to the enzyme. So we can put all this together in this model. Here's MTHFR, here's MTHFD1, taking tetrahydrofolate and formate to make methylene tetrahydrofolate, or methanol that goes to methylene. Here's serine hydroxymethyltransferase, taking serine to glycine tetrahydrofolate to methylene tetrahydrofolate. This is the enzyme that's highly polymorphic, and it's the gatekeeper making 5-methyl tetrahydrofolate. It goes on to make methionine, but also inhibits this reaction. When this enzyme goes down, what we see is this enzyme, and this enzyme become hyperreactive. Instead of having one carbons going down to methionine, you're converting glycine to serine. And we were able then to show in cells that these individuals who have this polymorphism not only have decreased rates in methionine synthesis, they are hyper synthesizers of serine from glycine. That's something we never would have thought of unless we had looked at this model. And so we're using this model to ask all sorts of questions and then going back into the network using isotopes to try to understand it. And there's probably a physiological meaning of this because glycine is a glucogenic amino acid. To make glucose from glycine, you can either glycine, serine, all the way up to pyruvate. And so probably this is regulated under conditions where you need to make glycine from serine, but those with the polymorphism clearly have hyperactivity in converting glycine to serine. The other thing we've done is modeled uracil capacity. I just want to close very quickly and ask the question, why does uracil accumulate in DNA? When we model that pathway and we don't have a nuclear compartment or try to constrain around a metabolic complex, what we can do, cells can only make 60% of the thymidylate they need. Yeast can make 3.6-fold above the amount of uracil that they need. We've now worked on this model to put in a nuclear component and indeed constrain it in a channeling model where the folate is never free to channel from enzyme to enzyme. And in fact, we can get the number up to about 1.2. And so we're iterating back between the cells in this model to try to see what the actual capacity is and why the million cells live on the edge in terms of how much thymidylate they have. We see very little impact on any rehab. Serine comes from glycolysis. We don't see a case where serine is ever limiting, where you can use serine as a push, as an input into the network. Glycine is different. In cells that use glycine, and there's just a limited number of the glycine cleavage system, you can push with glycine. You can't push with serine, and I don't understand why.
So, a related question. So, of course, the pathway also produces NADPH, right? So, is there any role for that in any of the pathways? I mean,
We're looking at that. We've stayed away from that. One of the problems, there was a paper in Nature a couple years ago that said that this pathway was a major producer of NADPH.
40% or something.
Yeah.
Some crazy.
Nobody's been able to replicate that. The laboratory can't replicate it, and they won't retract the paper, which is an issue with me, but I'll just leave it at that.
For the neural tube defects, there's of course neural crust
Yep.
involvement.
Yep.
And with cancer, a lot of the before cancer cells take on the stem cell characteristics. So have you looked at your pathway in specifically stem cells and stem cell division? Is it the heavy proliferation, or is there something about stem cellness that's
Okay. So what we wanted to do was ask the question, or a slightly different question, and undifferentiated cells are interesting. So if you decide that your readout is histomethylation, what you can find is not in a differentiated cell or a cancer cell, but in a stem cell, if you dose in more input in terms of a one carbon, it's either formate or covalent, you can get proportional increases in density of methyl histones in general. Same thing if you give them acetate. You can increase the amount of acetate, of acetylated histones. So we set up this screen, a 2x2 screen, where we looked at one carbon input on microturner plates as a function of acetate to see how they competed and looked at differentiation potential and everything else. The experiment failed because we rediscovered a paper from 1960. Serine was our main input. And stem cells, for stem cells, serine is an essential amino acid. I didn't know that, but it is. Stem cells absolutely need serine to grow. It gets back to your point. Differentiated cells do not. So we're going to go back and do that experiment sometime. This is a cool experiment. We have to totally redesign it because of that. But your question specific, that wasn't really
whether you
answered.
could separate the rapid proliferation from what we think of as stem cellness.
Yes.
Those markers.
Yeah, that was our approach to begin to look at this, to look at the programming effects of one carbon metabolism and whether we could differentiate them, whether we could lock in sort of that epigenetic landscape type thing. So we're thinking about that, but we haven't looked at it. We're curious.
How does fumonisin act in your system?
So we collaborated with someone, so fumonisin causes neural tube defects. We know now that it does not work through one carbon metabolism. It works through the PCP pathway, the planar cell polarity pathway. That's very clear. And that is about 10 to 15% of human neural tube defects. But that's sort of a hardwired, right, developmental pathway, cell biology pathway. It's not affected by folate. Does fumonisin affect this? Yes. It affects serine metabolism because of the sphingomyelin synthesis relates to serine and inhibits that. So you do see a perturbation in serine metabolism with fumonisin, but it's not related to the neural tube defect.
Do levels of U versus DU also go up in DNA? Because that's easier to look at.
That's a compounder we always have to deal with. We don't know the answer to that. We don't see that, but we're not good at measuring it. We have tried to check as a control. Does UNG1 excise uracil versus DU from DNA, which would be a real problem for us, and it doesn't seem to.
Okay, but if it's in there, you could directly look at it. So
Yes.
if DTTP was going down,
Yeah.
and you were getting both more DU and more RU,
Yeah,
you can directly look at RU.
We have not done that, and we should, or somebody should. Yeah, absolutely. That's a really good idea.
Would an MTHFR inhibitor synergize with the DHFR inhibitor? Or antagonize? I was trying to figure that out.
So, yeah, years ago, there was a big pharmaceutical push to find MTHFR antagonists. Never really went. They developed them, I know. I don't think they had any therapeutic effect. Don't know if they were used in any combination therapy, but you would think they might. Yeah, I don't know the answer to that. I know that they were developed, and I know that they were abandoned. I don't know if they were used in combination. That's a good thought.
I'm just wondering if the exporter for the one carbon unit mitochondria is known. If so, is it a major regulator for carbon units synthesization?
So the transporters, I think Sabatini said he has the serine transporter. I haven't seen that published yet. The glycine it's a serine glycine transporter, I think. The formate transporter is not known to my knowledge. People talk about that. I'm not interested in transporters, but some people are.
No, I was wondering, the reason why I asked this question is it a major regulator, or does it limit the amount of carbon that come out from mitochondria?
Oh, so, right, so the question is formate ever limiting, right?
Right.
Is formate ever limiting? And that's a huge question right now, because one of the things that we see is that when you decrease, MTHFD1 is what's going to capture the formate, right? If you don't capture it, it goes right into serum. So people have increasingly looked at this, because now we have good mass spec techniques to measure formate pretty reliably. Well, I don't, other people do. That in fact you see in folate deficiency in these MTHFD1 mutant individuals, and they are mutated versions, serum formate goes away. You know, is mitochondrial formate production ever rate limiting? People are working on that. So when we look at our in silico model, it's not. But that's, you know, that's just hypothesis generation.
I've got a question. I believe you said that folate is substrate or nitric to all the proteins that can bind it.
It's much, yes, it's 5 to
Is that
7
taken into
level.
account in your model?
Yes.
And does that affect the rate of responsiveness? Or, I mean, do you ever look at kinetics with regard to your model in terms of reshuffling the layers in response to?
Oh, absolutely, absolutely. We look at how that affects the partitioning. What we haven't done yet, I love the computer scientists I work with, because I don't know any of this stuff, and they actually are really good to work at. If you have somebody who actually talked to a biologist, and we get frustrated with each other, and the Italian doesn't help. But what we want them to be able to do is, there's really good evidence from Benkovic's work that purine biosynthesis occurs as a multi-enzyme complex in a purinosome. There's no evidence that the homocysteine remethylation enzymes are in a complex at all. That's just assumed. And then we know that thymidylate biosynthesis is a complex. And we want them to model, rather than diffusion, we want them to model these things as channeling, which probably occurs, right? And we know it occurs for some of the bifunctional enzymes in prokaryotes. And so they just rather take those kinetic variables, you know, and from the studies from the eukaryotes and just put in a fudge factor than actually build a model for channeling. But that's what has to be done. And it makes sense that, so if you do look at the model, we have done this, but this is almost intuitive. If these enzymes are in a complex and channeling the cofactor from enzyme to enzyme, you always need a free acceptor site or it doesn't work. So you have to be at least 50% subsaturated or you can't channel. And so we're trying to understand, you know, what optimal saturation is, and it's certainly not stoichiometric, but what that ratio is that enables channeling for some of the pathways, while the other ones may be dependent on diffusion and how you strike that balance.
Okay, if no further questions, let's thank the reception starting now.